So my name is Alex Schneider. I'm chemist. I make my PhD in Germany. I'm biochemist. I work since more than 20 years in science. So now I work in EATG. It's a university in Zurich as a scientist. And parallel, I am a program chair of Partner in Science by EATG. And today I will give you this presentation. So let's start. Exactly. So we decided to make this new training or a new module about production of HIV drugs. And as I told, this is usually a training for 1 day in the 2 different parts, in the morning and the afternoon. And today I will present you the first part. We have 2 hours. And from the experience from yesterday, we see that it's usually we can be a little bit even early with the finish of our presentation. And exactly. So let's start. We will talk about production of HIV drugs. And drugs for us, it's always drugs, medication. Usually we call this medicine or medical products. There are so many different names, and we always means that something what help us against something, for example, against HIV or against hepatitis C and so on. So we just say medicine. But to be honest, it's just not medicine, because this medicine consists of 2 important parts. So in one part, this is API. This is active pharmaceuticals ingredient, usually just normal powder. And you can see here's a picture. So it looks like just normal white, or sometimes it's yellow or different colors. It's just a powder. But this is API. This is if you understand the name, it's active pharmaceuticals ingredients. It means it's an ingredient, it's a substance which very active. So it means that this powder help us, for example, against HIV. It means this is exactly the substance or compound, for example, abacavir, lamivudine, exactly this powder, which inhibit, for example, integrase or protease in our body against HIV. So this is exactly this what's help us. And maybe someone remember, even I cannot say, maybe in 70s or in 80s, you usually got early in pharmacy, if you ask about some medicine, you've got powder. It would be like a weight, I don't know, like 200 milligrams weight, and give you very like in 80s, especially. I can remember, I'm not so old, but I can remember from Soviet Union times that we always get in pharmacy this powder, and it was just a compound which help you. You just take it, and I can remember as a child, it was really bitter. It was a chemistry, so you don't like this to take it. So this is why people develop the second part of medicine. This is called usually finished dosage form, and this is exactly what we usually now can see in the pharmacy. This is tablets, solutions, drops, etc., etc., etc. So this was developed in the past, because chemistry, it's the taste, it's bitter. So and sometimes it's also shelf life. If you have a contact with the sun, or if you have a contact with the humidity, your compound, your active ingredient can destroy with the time. And scientists say, okay, we need to cover, we need to put in a capsule, we need to put in a tablets, or we need to, for the children, it's better to have a solution because they easily can drink and so on. So this is how API was confirmed in such a finished dosage form. And this is important to understand, this both parts, because this is about the first part, about API. This is what we are talking today, will be our training. And the second part, we will talk about production of tablets and solution and drops and so on. So this will be our second part. So, and if you are speaking about medications, also very important to understand, do we speak about API produced by chemical synthesis, or just normal chemistry, we say, or we have an API produced using biological source? This is important to distinguish these 2 parts, because we will talk today about chemical synthesis. Biological source, I will bring you some examples because in hepatitis C and future in HIV, we will have such a medication, and maybe it will be the training for the next year. But for this year, we decided to make just only chemical synthesis. So here you can see some examples. What does it mean, API produced by chemical synthesis? This is, for example, sildenafil. Sildenafil, this is just normal Viagra, so you can see. Or our abacavir or imatinib against cancer, or dolutegravir. It's just, you can see it just normal, small molecules. So you can always, if you look for some medication, you look in Wikipedia, you can always see, if you see such a structure, which you can clearly see that you have some, I don't know, some elements, and you can calculate it that it's not so many elements on the picture, it's everything put it on the picture. This means you have a small molecule. It means usually you get it by chemical synthesis. And if you have such pictures, so it means that something very complex, very, very complex. So it's erythropoietin, or for example, monoclonal antibodies, which we are going to use for HIV also, or for example, blood coagulation factor number 8, or interferon alpha 2b. I put it here because this is, we still use in hepatitis C. And this is interferon. It's produced using biological source. And biological source, in this case, usually you have a bacteria, and you put via virus special gene. So, but we will not explain, I will not explain more about this. I just want to show that the differences about the small molecules. For example, aspirin, you can compare with the board. So it's like a 21 atoms. And then you go to the small biological molecules. It's, for example, somatotropin. So it's about 3,000 atoms. So you have a bigger board. And then, of course, you have I don't know, AIDA or something like a cruise, which now not go through the sea because of Corona, but you can see the complexity. So it's, if you go from the small molecule and we go to the bigger molecule. So, and this is, of course, if some molecule has a more complexity, it means also in the production, you have a more complexity. So this is, I will bring you today only about the chemicals. This will be enough for us against HIV nowadays, but for the future, we see it will be the needs that we will need to explain how to produce biological and to be more aware about this process, because if you're aware about this process, you can use this in your advocacy work, because you understand how the medication can be produced, and you can really be a partner in negotiation with the pharmaceuticals, for example, companies. So we'll talk today about production of API, and tomorrow, on Friday, we'll speak about finished dosage form. And as an example, I took HIV drugs. Why we use this, training? It starts about 5 years ago, because at that time, in the earlier 2000s, so after that originators, we have on the market originators drugs, like from Roche also, and from ViiV, from Janssen Cilac. At that time we didn't have a Gilead, but later Gilead. So, and later generics were coming on the market, and we as an expert, I work in the chemistry since 1990s, 95. So of course I understand what's the difference between generics, between originators, so on. But many of our community partners didn't understand that generics are coming, and for them it was something new. And we know that, you know, it's from stigma and discrimination of HIV. We know that if something new it comes fear. And fear, it comes to ignorance. So, and of course, it's in the case of generics, happens the same one. And we've got many myths. So many myths about generics. And here I just can show you, so from professional community, even from the doctors and from professional community association, we could hear always that the generics, they have a poor quality. The generics, it's ineffective. You cannot use them. So, and generics cause additional side effects. They're cheap and it means that they're bad. So this is what we can hear from professional community, and from our community, we speak usually the normal language, easy language, so like generics can be poison, generics can stay not tasty, don't help, and I feel sick after generics, and flu and paper are added to generics. We can hear since many years about this, and this is why we decided that we need to start such a training about production to explain our young advocates that they understand, and all of these myths can be dissolved, because they will understand and they will immediately see the difference between generics and originator and can understand what means poor quality, if it's because of generic or if it's because of production process. So this was the idea of this training. So, and we will go today and on Friday through all of these points, because they will help us to have a good structure of our training. We go there like check each mythos and explain if it's still mythos, or we can understand that it's in the reality it looks other. So I will start with 2 points at the start that usually we say that generics quality is poor and generics are ineffective. And to understand these 2 points and to have an answer, we will start with again with the medicine, and we will speak about production of API. And as I told you that medicine consists from API, very important point, and from finished dosage form. And if you look, because sometimes it's difficult to understand for the people, they cannot, if I say that this is production of API and this is production of finished dosage form, they look for me and say, okay, you studied 5 years and then you make a PhD. Of course, for you, it's like obviously. For us, it's not obviously. We need to we have a visualization, and I can understand you. And here's the visualization. So API, it's a normal chemical plant. And finished dosage form, this is pharmaceuticals plant. And you can see the picture that it's a different picture, because if you are talking about APIs, this is just a compound. It's just a compound. It's not tablet. It's nothing. It's just a powder. It's just abacavir. It's just I don't know, sofosbuvir. It's just this compound. This is a structure which I show you. It's only this one was produced, and this is produced in a chemical. It's just normal chemical plant. And then from this chemical plant goes to the pharmaceuticals plant, and in this pharmaceuticals plant it makes tablet or make solution or put in the capsule and then sell to customers, to you, so or to us. And pharmaceuticals companies, which you know is usually like a ViiV, Gilead, they don't have a chemical plants. They don't have it. They have usually just a pharmaceutical plants, and even not enough then. So they have like a know-how, they have a patent, they go to the chemical plants. For example, here in Switzerland, we have 4 different big chemical plants where Roche, Novartis, Janssen, AstraZeneca, everybody goes to these plants and say, okay, look at, you have a chemist there, you have facilities. I would like that you synthesize this molecule, for example, abacavir. And this is just normal chemistry works in this chemical plants. Today they do for Janssen, I don't know, Durunavir, and tomorrow they do, I don't know, something for the oil production or for the plastic, and so just normal chemical plant. So, and in pharmaceuticals plant then goes there, and pharmaceuticals plant they have also contract with the pharmaceuticals company, or most pharmaceuticals plant already like belong to pharmaceuticals companies, and they produce already like this finished dosage form. And in our case, it's just normal tablets. You know that most of them, we use just normal tablets. So how to recognize chemical plant? It looks like this. So you have big columns. So you have a steam at sticky. So it always you go and you see that it smells not so good. It smell. I mean, for me, it smelled like my studium time. So it's still good smelling for some chemist people, but I can understand for you it smells not so good. It's like just normal chemistry. So sometimes it looks like a petroleum factory. So if you have seen in your, I know that in Poland and in Ireland and in many countries you have this petroleum factory. So it's exactly the same. It's the same chemical plant, because in petroleum factories they do the same. They also make a different chemistry processes after they separate oil. They do some chemical processes. So and then they sell this intermediate to other chemical, for example.
Alex, I have a question, quick
yes,
one.
Yes,
so sometimes when we are ordering our HIV medication from pharmacy, they needed to order it from somewhere else. Do they order them then from chemical plants, or it is another space?
I mean, they usually order anyway from warehouse. So all of pharmaceuticals lager on the warehouse, and warehouse they take this our tablets. They take finished dosage forms. They take already from pharmaceuticals plant. They don't take from the chemical plant. Chemical plant, this is only for production APIs, and this is only for to make later finished dosage forms. So this is only for pharmaceuticals companies. So if why I explain you this, because in the second part, we will speak about pricing. We will speak about how much does it cost? Because usually always if you speak with some pharmaceuticals company, say, look it, you have a great drug, but the price for me, it's unacceptable, because it's not possible to have this drug for all our patients. And then pharmaceuticals companies start to clarify it's a production process. It's so complicated. It's so complicated. It costs us 100 millions. And if I hear this, sometimes I just laugh in my side, because it's not true. Because usually production, and we will see it in the second part, it's for them, it's less than 10%, all of this cost what they at the end sell us. And this is why I try to explain you these differences, that you understand that chemical plant make this compound, sell to pharmaceuticals companies, and pharmaceutical companies make on the pharmaceuticals plant, make the tablet, and then sell this tablet to us.
Alex,
yes,
I have a question. Could it be that, based on what you say, that the generics and the original drugs are basically coming from the same chemical plant?
Yes, this is you just take words. Have you seen my presentation before? because you take my words from my mouth. So exactly. So this is what I want to explain. That, for example, if we are talking about lopinavir, ritonavir, everybody knows this Kaletra. And I can remember that we had a huge discussion, especially in Russia and in Ukraine, about this production process and the generics and so on. And in the world, you have just four factories, and all of it's 3 factories. It's in India and 1 factory in China, which make lopinavir as the API. And AbbVie, Cipla, all generics and originator company buy there. So at the end, you have the same everywhere. At the end, it's just how the pharmaceuticals companies makes later the tablet. And we know, for example, in lopinavir, ritonavir, Kaletra, we will speak also on Friday, there is some tricky production process, because production process of pharmaceuticals product, finished dosage form, it's also not so easy. It's not just press the tablets. There are some tricky. So we will talk on Friday, and you will see that could be here also not so easy to get a generics complete copy. But if you speak about normal tablets, like I don't know, abacavir, which really pressed with or aspirin or something like a paracetamol, so there is no any differences. There are several factories, and why we have like a few amount of these factories. First of all, because there is no like a big demand. If we are talking about HIV, it's not like a paracetamol, which everybody, or ibuprofen, which everybody takes. So you have like a billion people which takes this, and you have a huge amount which should be produced. In that case, you have like 100 chemical plant which produces ibuprofen. If you're talking about HIV drugs, usually you have a really few plants which make this synthesis, and they make immediately, because if they make in a huge amount, it can bring cost effectiveness, so it can bring cheaper price. Of course, if I do, this is why in Russia they try always to make independence. Even now they try to see that we make a Russian product, but it's not Russian products, because they take API from outside, from abroad, and they make it just a tablet, or some factory make it just packages, and they say, oh, it's a Russian product. It's not Russian product at all, because it's not just to make for one country. If you're talking about, you know, Switzerland, or talking about Germany or Poland, it is not enough amount. If you do it in the chemical, it will be too expensive. So this is why you have like a few amount for whole world, and this is enough. And this is why we'll talk about the pricing on Friday. You will see how much does it cost, all of this production process. So let's talk about, again, chemical plant. I have here some picture from inside. So a chemical, it means that you make a synthesis, and this is usually you have also many reactors. You have many like such a synthesis volumes inside. And this is like also the picture from one chemical plant in Russia, because I made an audit. I mean, you can see someone who sees this picture and say, oh, it's so dirty. Yes, it's dirty. But you know, chemistry, it's always dirty. Chemistry, it's always smell. It's always dirty. It's normal. It's very important how this is inside in this vessel. And then inside, it's very important that it's a clean. But outside, it's, for me, it should be, if it's too clean, it means that they waited that someone come and make it everything clean, you know. So it's this is not true in chemistry and in chemical plant. Even in Switzerland here, where you have a really strict regulation, if you go to the chemical plant, it looks like in a chemical laboratory. So it looks not so very clean. So it's normal. So, but if you go to the pharmaceuticals plant, of course, you don't see any tubes on any columns, any steam go out. It's just like a building, and inside the building, it's clean. It's like you will do now surgery. You know, it's everything clean. You have a filter, HEPA 14. It's no microbes, no that, not that, not that. So here, it's a really, really strict condition. In a chemical plant, you just make your substances. And then to put the substances in finished dosage form, it's very important to be clean, because this finished dosage form you take inside. So, and this is why it's important here to make a production very clean and to have a very strict condition. Also in a chemical plant, usually you have like a last stage. If you produce, for example, API, at the last stage, usually you should have also such a filter and a clean room, because the last stage goes to the pharmaceuticals plant and should be already clean. So, and in a big chemical plant, you have usually one such a room, only one, where just make every package, make it clean, and send to the pharmaceuticals company. So this is very important to understand, because usually you see such pictures. If you talk with the pharmaceuticals company, you see if they explain how they produce, they show you these pictures. But this is how they produce the finished dosage form. This is tablet. But for us, very important, the substances, because substance help us against HIV, not the tablet. The tablet is just amount of different substances together. We'll speak about this on Friday. There are many inactive substances, but only one active substance, which very important for us, which help us. And in these substances, important to have a good quality, to have a good production, and to don't have a side effect. So this is what we'll talk later. So I have here just one video. I don't know. So you can see that it's, because usually pharmaceutical companies, they would like to show like such videos to see that how clean they work, and they usually show this machine which just make packages of, you can see as a blisters, the tablets, so it's just a repackage of production process. But it looks very nice. It's what you can present. And usually, if I can remember at the start with the community, we see this video and say, oh, so nice, so nice, so nice. But at the end, it doesn't matter how this machine works for us. It's important what's inside you have in this each tablet. So, and again, so we have an API and we have a finished dosage form. We will talk today about API and about chemical plant. So about synthesis and production of API. Why it's also important? Because if you, for example, if you, maybe I will speak a little bit already today, and on Friday we will repeat this. If you want to know, for example, where your, not tablet, but for example, where API comes from, and you would like to find out. So this is important to know this word API, because if you just put in a Google like API and then abacavir, API and then sofosbuvir, you will find already many, also in Alibaba and everywhere you can find that you can buy all of this API in a kilogram, or you can buy in a ton and so on. So this is like pharmaceuticals companies works, especially generics. They just look for the plant which produces API. So, and you can always, even you as an activist, you can just ask this plant and ask how much does it cost, for example, if I buy 1 kilogram of API. I can remember the time of sofosbuvir. Now you can buy sofosbuvir generics without any problem because there is a buyer clubs and so on. But 5 years ago, I can remember the time we didn't have this buyers clubs. And myself, with other guys, we go to India. I hope that you know, explain to someone about this story. So we go to India and we buy there just 1 kilogram of sofosbuvir. It was just a white powder. So it's on the border, someone will say, oh, it's a cox. No, it's not cox. It was the sofosbuvir. We bring this sofosbuvir to other country. And I took this sofosbuvir. I have a balance. And I just, okay, we need so and so many milligrams. I put in the capsule, the next one, the next one, the next one. So, you know, because at the end, it doesn't matter. It's important that you have a sofosbuvir inside, and the sofosbuvir help you against hepatitis C. And so we could help so many people at that time because it was incredibly expensive to buy, and you could not buy via buyers clubs at that time. Now it's good. You can buy via buyer clubs. But I can say that this is API very important. So even if you would have this powder of, I don't know, for example, you have a Truvada, tenofovir emtricitabine, you can just take with a spoon and eat each day, and it will help the same as help you use the tablet. So this is, I just try to explain you that this powder, this API, it's very important for us, for the activists to understand, and especially if you look for advocacy, look always API and then your medication if you are interested for one medication. So go on. So
question. Yes, question.
Yes, Alan.
So all HIV medication, do they have also API? We can have API available.
Yes, yeah. API, it's a general, each medication has API, each medication. So each medication has an active pharmaceuticals ingredients. Some medication, for example, if you're talking about Truvada, you have in Truvada 2 APIs. You have one API, it's tenofovir, and the second API, it's emtricitabine. So you have 2 active ingredients in one tablet. So, but each medication has an API. So, but for example, if you would like to find out how much does it cost, you put Truvada, you will find how much does it cost Truvada. But it's for you, it's not interesting. You would like to know production cost. So in this case, I would suggest you put tenofovir API. Look for the price tenofovir as an API. Then look for the price emtricitabine as an API. You will find in the internet. I will show on Friday how does it work. So you will find very easy each tablet, and we can approximately calculate how much does it cost the production. So here I would like to, what does it mean always if I speak about API in case of HIV drugs?
Ask you one question, Alex. I'm sorry.
Yes, Peter?
When you're talking API, you are talking about your small canoe. I mean, it's not about the cruise ship or the big sailing ship, but BNAPs is not API that is developed in a pharmaceutical, in a chemical plant.
BNAP.
Yeah, sorry.
BNAP, BNAP. Yeah, okay, yeah, I understand what you. No, I mean, this is also API, but it's this API from logical source. So we don't about biological source. We speak now about APIs which are made by chemical synthesis. So, and
exactly.
Nowadays, here's an example. All of our HIV drugs, they have just normal APIs from chemical synthesis. So if we are talking about APIs of HIV drugs, and this is a good question from the Peter, for us, it's always the small molecule. It's always like, you see like here, abacavir, zidovudin, lamivudin, tenofovir, emtricitabine, and so on and so on. This is always small molecules. You can find this structure, chemical structure in the Wikipedia everywhere, so it's not so difficult to find this. And you can see here, for example, I mean, if you don't see something, it means that you have a C, that you have a hydrogen and so on, but it doesn't, we don't need to understand now. So it just understands that if you see the structure, clear structure, so it's usually 99% that it's made by chemical synthesis. Even such a structure, and enfuritidine, for example, from Fusion, it's from Roche. So it's a Fusion inhibitor. So even here, we still have a clear structure. So it's a clear structure. You have many amino bonds there, amino acid bonds, but it's still made also by chemical synthesis. So, and if you're talking about, for example, antibodies, so this is everything what's made with biological sources. There is a complete different process how they produce them. So, and they also say that it's an API. It's also API of this, for example, drug, or for this antibody. There is also API, but there are some differences, and we will not speak here because for HIV drugs, for us now, it's not important to understand these differences in these biological products because we don't have it. In the future, we are going to have it, and in the future, I think we will bring this. Even if you're talking about hepatitis C, we still have it interferon in hepatitis C, which make by biological source. But you know that everybody tried to get sofosbuvir, and I explained you already how easy to do it with the powder, and you will be a cure from hepatitis C. So I don't see the also importance for advocacy to understand how we produce interferon. So this is why we don't cover these points, but I can show also what I explained now about API of hepatitis C. Now we have these new APIs or new drugs, all of them made by chemical synthesis, for example, sofosbuvir, daclatazine, lidisbat, and so on. All of them, you see that it looks like complicated, but you still see the clear structure. So, and if you're speaking about biological source, here, ribavirin, for example, it's also chemical synthesis, but it's not, it's complementary, it's not against hepatitis C, but against hepatitis C, it's interferon, for example, alpha-2b, and here you can see it's already not clear structure. It's already, we try to show biological structure. We try to show some biological product in some structure. So, and it's usually looks, here it looks like a picture, and here it looks more structure, more like plain bonds, and you understand, okay, this is something chemical, and this is something biological. So you can see even in the Wikipedia, if they don't explain how this compound was produced, you can already from the picture understand if we are speaking about small molecule and about chemical synthesis, or we are speaking about big molecule and about biological product. So let's go to the synthesis, and let's go, I just take as an example abacavir, but it doesn't matter. We can take lamivudine, we can take all of these APIs which we have in HIV now. So there is a chemical process, and chemical process, for example, now it looks like this one. So if you want to get abacavir, nowadays it works like this. So, and if you see, first of all, you say, my God, yeah, it looks like a crazy man put something on the paper. Exactly, some crazy man put it on the paper, but it's still understandable that we try to get from the compound number 6, there is a 2 parallel lines. We try to get compound number 11, here compound number 14, 12, 15, and so on, so on. Then we have these 2 compounds. We put all together, we get Z1 and so on, so on. And at the end, we have compound 21. And this compound 21, this is our abacavir. So you can see, oh, why is it so complicated? Because first of all, our molecule, it's not so easy. You see that it's also, it's a big molecule. You need to build these molecules. You have this is like a different building blocks. Different building blocks you put, it's like sometimes looks like a Lego, which building blocks come together. And in these building blocks, you build your molecule. And you usually start from very, very small and easy going molecule. In this case, it's a number 6. And this molecule, you can get like a very cheap from just normal oil, petroleum production, so from oil. And I can tell you that our background sources for all medication, this is oil, coal, and gas. Everything done from this, everything. So this is like you have many syntheses, but at the end, all of our tablets, this is just from oil, most of oil, you have a gas and coal. So this is why you have many syntheses, and then you have abacavir, and this is how this chain works. This is the process number 1 I called, and then we go, there is also alternative process number 2. In this case, this process was at the first, should be like in process number 1 and process number 2, it's now other one, but I just put it 1, 2, it doesn't matter. So of course, I just would like to explain you that to get abacavir, it's also like a Lego. You can build the one exactly house, of course, from different building blocks. So the same it's here. To get this molecule, you can start, for example, from this molecule. Here start from this molecule, and here start from number 22 molecule. So it doesn't matter. I mean, it's just, this is exactly what chemists make. For example, I am chemist. Pharmaceutical companies come to me and say, okay, I would like to have abacavir. And then I look abacavir and say, okay, abacavir has a different building blocks. Okay, I know that these building blocks can be come together with this reaction, this with that reaction, and and I go from big molecule, try to put from which building blocks I need to start, and at the end I come, for example, to number 22 or to number 66 in this case. So it means that this is maybe important already for advocacy. If we have a new molecule, for example, abacavir. Of course, first of all, the pharmaceutical company start to make a patent for this molecule. So in this patent, it goes for 20 years. And of course, in this case, it's a patent the molecule as it is against of HIV. It means, you cannot do nothing because they just found out that this molecule help against HIV. What pharmaceutical companies also make parallel, they make always new synthesis process. They say, okay, look at synthesis. This is too expensive. Let's do it a little bit cheaper. Let's do it that. They always improve themselves, and they make a new process. They make again new, and all these processes. For example, this is process how abacavir works now, and this is process. It's a patent process, and they make this patent later, because first of all, it was the second process, and this is this process, it's also patented. So, and sometimes you can hear also from the pharmaceuticals companies, if you go to the generics company and say, look at, why you don't do it abacavir? Because abacavir, it was patented in 82, 20 years until 2002. The molecule, it's not patented. You can produce? And they say, pharmaceuticals, sorry, we cannot, because there is a different synthesis processes for API, and the process, which very cheap and cost effective, usually it's also patented. And then they just wait until this patent expired, and they can also produce. So it was exactly with abacavir, or for example with darunavir. We still have several APIs which you don't have a patent for molecule, but you still have a patent from process, and many pharmaceuticals companies still have this barrier to produce API.
Alex.
Yes, Greg.
This is very interesting. I was not aware of this. Can you a little bit elaborate on the timing for these patents? Are they? I assume they are overlapping to some extent. I understand that the patent for the substance for the API is 20 years, right? What about for the process? Is it also 20 years?
Yes, and it's I mean, this is extension. It's what exists. This is not on the patent, and this extension of the patent there is only in the US. So and in Europe it's only 20 years. And for example, look at, I have an abacavir. I cannot. It's just example because I don't know exactly the year. But for example, I found out that abacavir helped against HIV 1980. So I immediately make a patent because I need to protect myself. Yeah. So it means I have a 1980 plus 20 years until 2000. Mm-hmm. What's the company make? Because they just found out, and they start to look if it really works. And then they say, oh, it really works. It took like 2 years. And then like a third and fourth years, they start to find out how to produce this molecule, not in laboratory, in a big amount in the production. And then they found out, oh, look there is one process, and this process, it's unique. Let's make a patent. So, okay, it's like 4 years later, 1984, they have a new patent until 2004, only for the process. Okay. Then, like always it takes from the molecule discovery until they go to the market, it's usually 10, 11 years. So it means 1991, this molecule will be already as a tablet, approved, and you as a patient can take it. Mm-hmm. And already at this moment in 1990, they say, okay, look we have so many patients, we need to make more production. But this chemical process, number one, for example, number two doesn't matter. It doesn't work very well because we need a more amount of our API. They find out the new process. In 1990 or 92, they make a new patent until 2012. And and then they say, for example, okay, look at this dosage form, which we make this tablet. This is always, I don't know, you need to put in a refrigerator. For example, it was with ritonavir. And they say, let's find out the new form that it's not you need to put in a refrigerator, that you can keep on the room temperature. The same API, the molecule is already discovered 40 years ago, but I make a heat resistance tablet, make a patent. So 98, I have a patent until 2018.
But this already sounds to me like some kind of a play, and this sounds to me very non-compliant.
Of course, I mean,
is there any process to control such behavior patterns? Because that's obviously made for money. That's not.
Of course, sure. I mean, to be honest, I mean, sometimes, of course, if there is, for example, one process, there is an Adalat, one product, it's against of pressure, so blood pressure. So, and Bayer, they make, this is a product to decrease your blood pressure or to control your blood pressure. There are many like APIs. It's also like from 50s years production. But what they make really the tablet so complicated that during 24 hours, you take from this tablet so small amount that it's you have like your blood pressure always under control. And they patent this tablet. So this is, I can understand, this is really value added because they find out how to make this, like how to deliver your medication.
Like slow release or something like
that.
that.
Yeah, exactly. Oh, yeah, good. Yeah, slow release, exactly. So, but in many cases, for example, what we see with lopinavir-ritonavir, this is so old drug, but it's still patented in so many countries. And this is like unacceptable from AbbVie what they do it, because this is why community, and this is we do such a course that you understand more and more, and all of this chemistry knowledge you can usually use after that if you make against such a patent as a paper or something like that. But there is no any institution which monitor a patent. You have just normal patent authority which make a patent, and there are of course different legal situation in each country. In several countries you have such a possibility of green patents. It means that it's always make some small differences. Oh, something new. Oh, something new. And at the end, it's nothing new. So, and there are some, like in Europe, they always try to make a strict legislation of the patent that all of these green patents not coming, but it's still there. So, and you're right. In this point, we as a community should monitor and try to bring them, or try to make a pressure, just to try that they understand, that we understand how they produce, and that it's just bullshit what they explain us about something new, and that they spent millions for this production, for example. So, okay. But I don't know, do you have some patent opposition in your course? I think you will have it. I think you will have it. So, and I think you will, or I don't know, Peter.
No, I don't think we have that in the program.
I know you don't have it. Or maybe we need to speak about, because
yeah.
I mean, this is the point which at the end, what we should bring to our advocates, that they use all of our knowledge also in the patent oppositions.
And I was actually going to ask you a question, Alex, with your knowledge and you knowing how these molecules are being developed. What do you have an example of bullshit?
For example, yeah, lopinavir-ritonavir. It's I mean, this is the old molecule which already should not be patented at all. So
and who should be advocating to then?
No, exactly, and this is why we started, especially during COVID-19. This is why we put also from Switzerland. This is pressure to the AbbVie, and they make a patent like.
And is there? I'm sorry to be this direct, but is there any bullshit from our donors like Viv, Gilead, Janssen, MSD?
I mean, sometimes I mean, you cannot say that there is a. For example, we found once from Viv, it was a bullshit. In the pricing in Russia, it's also like maybe 7 years ago, they have like a abacavir, I can't remember, like a €1, lamivudine €1, and together it was like a €5. Would you say? I cannot understand why it's €1 plus €1 in one tablet. It costs more money, and they try to explain that it's to put these two molecules in one tablet. It's too complicated. What was really bullshit. So because this Quevexa was so easy to of course I would like to have a one tablet instead of two tablets. So, and then we make but in that case we work with the anti-monopoly agency, Kartellamt, and we could bring them to Or for example with the tenofovir and emtricitabine with the Gilead, we have the same problem. The same problem with the green patent, and this is also because of community and the pressure of PrEP community, especially in France. We've got so many generics, and we've got in Germany and so on. but we still have no generics, for example, in Russia. So we don't have enough pressure in Russia. So this is, even in our donors, we still have a bullshit, not bullshit medication. Medication, they have a good, but bullshit what they try to explain us as an explanation why it costs so, or why they have a monopoly.
Okay, that's great. I mean, I want a private course from you on this.
I am already a non-grad person for pharmaceuticals companies, so I still have a good audience even here. Okay.
Alex, question. Do you then tell them also that is a bullshit, just openly and discuss?
No, of course. I mean, the problem, Len, it's in the pharmaceuticals companies, they have so many managers, you know, managers to make an appointment. You know, it's like sometimes it's so difficult to get to the vice president or to people who really understand and to whom or with whom you can negotiate. Because as a community, first of all, you have some community person, and then this community person gives your information to the medical doctors or to affairs process, and so to come really to the person who really understand and can negotiate with you. It was, for example, as we start with the compulsory licensing in Russia and make a pressure to Gilead, it took like 1 year until we really get the contact to the vice president in Gilead in San Francisco, with whom we can really negotiate and say, okay, look at this is what you make here. It's a bullshit, and this is patent. It may not be on the market in Russia. So, but you can speak open. It's just you need to find the contact person which really understand and still be in company. Because I can remember with, for example, with the VIV, we have make a negotiation, very good negotiation about one product, and then this person just change the work and come to the other company, and then come new one, and you need to start again. So, and this takes a time. So, but with a community, if you have a support and you're not alone, it makes easy and strengthens you. So let's go to the next point. So here's just to show you as an example of Lidiposphere, also the hepatitis C. Also, you see a clear structure at the end. Also, we start with a small molecule and synthesis, many syntheses, and at the end we have a Lidiposphere. So we start with the oil, and we have a good drug against hepatitis C. So how we do it? All of the synthesis you have seen already on the other pictures. You have a chemical reactor, and in this chemical reactor, it looks like a pot. It looks like a pot. It looks like I would like to have a soup, you know. So it's for the soup you need the potatoes. You need here is the same one. So just to understand, and it's. I think it's also normal that always chemist people will be compared with cooks. So because you always say in the laboratory, oh, I go cooking. It means I make some synthesis. So because it looks like a cooking. So to make a soup, it's the same like to make a tenofovir, for example, or abacavir. You have a different reactants. You cook. You have sometimes you need for example, here you can heat or you can freeze or cool. For example, there are some reactions need to be at 0 degrees or at -10 degrees, even lower. Or you have some reaction at, I don't know, 80 degrees, should be boiling. So it's always the same like in the kitchen, what you do with your preparation of the food. So here's the same one. Here you can see this is like how look at inside and how it look outside. Again, you see that it's not so clean, but it's about exactly important that inside of this reactor it's very clean, and inside it's of course it's usually a closed system. You have many pipelines, and you can see here also it's everything everything what goes to your reactor, it's always pipeline. It's everything close, so it's go. So you don't have so if you can even make some reaction under nitrogen without any air. So it's also possible, and there are some reactions which you need to do it because your intermediate make a very quick oxidation on the air. So, and you make some, of course, at the end, your product should be stable because all our tablets at the end, it's you have an air. We are not on the moon, so we live on the Earth, and we still have an air. So but sometimes some intermediates, and intermediates it means here, for example, some like a pre-product, not at the end, not lidiposphere, but for example, this one it's already intermediate, or that one it's already intermediate, and several intermediates sometimes can be oxygen sensitive. So it means you need to make, for example, this stage you need to make under such a condition without air. So you put, for example, nitrogen, it's very cheap nitrogen. So in a laboratory, we make it with argon because institute and university has the money, but it's expensive and it doesn't matter if you make argon or nitrogen, but in the production, you make a nitrogen, it's very cheap because in our air, we have more than 70% of nitrogen, and to take this nitrogen from the air and put oxygen out, very easy process. So there are so many chemical plants which do that, and you get this nitrogen very, very cheap. So, and you can see also here, for example, you have a pipeline, so which bring your reactant, and there are some pipelines, for example, this one. So here you cannot see here like this one. So it means that you can put here either steam. For example, you need to bring your reactor to 100 degrees. You put the steam, so it's really hot, and put here in and here out. So it means you heat. Or you put some here, for example, coolant. So it's like a free, so like a refrigerator, and you try to cool down, for example, your chemical reactor. So, and here it's like all of these pots, or I would say, or chemical reactors, they have always two layers. So, and between these layers, you can put like a steam, coolant, and so on. So this is not a normal pot which you have in the kitchen. So it's like a little bit advanced pot for advanced cookers. So you have here API production. Here you can see the reactors. Usually, if you can imagine, you have seen that it's a big facilities. The reactors, for example, these reactors, which you can see on this, it's a 30,000 liters. It's like you have 3 floors. So you have on the first floors, that where you put out all of your product or solvent. There is a second floor with windows where you can look what's happened inside, and there is a third floor where you can put and look from the up, and here, for example, you can steer and so on. So you have, for example, here you have a steer which you can steer your reactant. So it's a really usually huge reactors, huge pot with so many pipelines, steer, and so on, and you have different levels. But very important what's happened inside. And inside it should be clean, and inside happened reaction. So here I have a small videos. I mean, it's show us how we make formic acid, exactly. So you have also here like chemical reactors, you have a steer. So you have you put sodium format, you put sulfuric acid, you have 2 reactants. Now you see we make a heating, we put steam, and our molecules start to moving, and we have a reaction. So this is what's happened. This is what's usually happened also in a laboratory. And here you have like 100,000 liters. So then you take your formic acid away. I mean, it's not like here, Larry doesn't come inside and you clean. It's just an animation. People don't clean from outside inside. So it's just animation. There are special clean processes, but just to understand. So it's really like cooking some soup. So this is like looks like chemical synthesis and chemical reactors. So let's go again to our Abacavir synthesis. We talk about that we start with, for example, here with the second 2 compound, and we have like a 1 chemical process, 2nd chemical process, 3rd chemical process, 4th, 5th, 6th. So it means you have 6 stages until you come to your Abacavir. And these 6 stages can happen in one factory. For example, you can make one reaction like here in blue, then the second reaction in yellow, the third reaction in white, and so on and so on. Or sometimes you can make some reaction in this factory, then you bring intermediate, you make in this, you bring here, and so on. So, you know, each chemical factories has a different amount of chemical reactors and different volumes. Sometimes you have a really big, smaller, because sometimes you don't need it so big. Sometimes at the start you need, even if you would like to have a 3 or 1,000 kilograms of your Abacavir, but at the start you start with a small amount, and then you, with the building blocks, then you at the end you will get 3,000, but you start with 30 kilograms. So it means that at the start I need small reactors, then I need the bigger, and at the end I need the biggest reactors, and sometimes even I need a really, I mean, each reactor has at the end limit of capacity. So about like this about 30,000 liters reactors. This is the biggest reactors which we have nowadays on the Earth. I mean, of course you can make a bigger, but it's in that case it's so complicated to make all of these processes inside of the reactors that it's not cost effective. So the biggest one, it's 30,000 liters, and from 30,000 liters, you can get at the end 600 kilogram of API. So, and if you look, and maybe you have seen already, sometimes in the tablet, you have a batch number this one, batch number that one, batch number that. So it means that exactly this batch, it's what's happened, it's what you can make in once. And API in once in a big amount without mix of different APIs, you can make a maximum 600 kilogram. So it means 600 kilogram, it will be one batch. This 600 kilograms maximum, which I make from Abacavir, then I go to, for example, to pharmaceuticals companies and make a tablet. And each tablet has for example, 300 milligrams. So, and then you can calculate it how many tablets you can make from these 600 kilograms. And it means so how many packages you can have in one batch. So, and then if I make again the synthesis process, this will be the second batch. If I make again, this will be the third batch. And this is very important to have a different batches, because if at the end in production, or for example, some patient will find out that, oh, suddenly I have some side effects, which usually you don't have it from this medication. So then I can really follow and find out, okay, it means, okay, in this batch, something happened. It means during this chemical process, something might happen. So, and this is why usually you make only one batch and you make a batch by batch and you follow this. Because if of course you can say, why I make in this reactor at the end 600, in this reactor at the end 600, and at that reactor at 600. I have a 3 times 600. I mix everything. I have already 1,800, and I can make it as a one batch. In that case, it's really difficult to follow and to find out the problem, what's happened. So this is why usually you make from one process from start until the end, it's a one batch. And at the end, you can use the biggest reactor, and at the end you can get maximal 600 kilograms. So this is why you can always calculate it, how big your batch might be at the end of the production, if someone interested and sees the different numbers and what does it mean. So let's talk about, again, synthesis, and again about Abacavir. We have one processes which I show you, which it's now patented and used from VIV to get this Abacavir in reality, this is real process. But you remember I told you that before that processes, we have this process. And you see that we have calculated here, you have 6 stages. I mean, we can, very easy to see that here 6 stages. Here you have 1, 2, 3, 4, 5. You have we don't need to calculate, but you can understand that here you have more than 6 stages. And then I say, okay, it's something wrong. It means this process with the more stages cheaper as this project with the 6 stages. Something does not fit. And you're right. So this is process. It's very, very expensive because at the end we have a chiral chromatography. And this is a stage. You have usually always like a last stage of the process at the start of always each molecule in the HIV production, because at the start you would like to get your product as fast as possible. For you, the cost of production doesn't matter because you would like to bring this product on the market. You would like to bring something against, for example, HIV. And then you make like a chromatography, I will explain later. This is like one separation process because you can see here and this molecule, I mean, you can see this is all these 2 molecules, it's identical. They're identical. In this case, I say, what they make, what is the separations they make. But then I see, for example, here, I have this bond. You see that it's like a bolt. So, and this means, I will show you later, this means that this is the molecule. This is the molecule which doesn't have any stereochemical, in the 3 days, there is no any position. And then with the chromatography, I separate different position and I take this position. If it's a bolt, it means that it looks like this one in a 3 day, and it's like it's still be this fixed angle. So, and I will explain later why it's very important. So, and but on this stage, very important to understand that at the start, all of our products in the 80s and 90s, even now, if they, for example, this doravirine from MSD, they still produce with a chiral chromatography because at the start you need to separate. And then you find out the way, because you see, for example, this process, they start already with the building blocks here at the start with, for example, 14 and 15, where they bring already stereochemistry. It means at the start, they bring already exactly this molecule where this OH goes up, you know? And here they just bring the molecule mixture and separate. This is from chemistry, this is easy way. But if you calculate money, this is very expensive. So this is why at the start, usually the process very expensive. But then with the time, API goes cheaper. And if you can see and monitor how much does it cost, for example, sofosbuvir, as we bought 5 years ago, and now it's already 10 times cheaper. So it's always, the process always developed itself and goes cheaper and cheaper. So this is why if you talk with the pharmaceutical companies today, and we'll talk and make advocacy in 1 year, you can with 100% know that now process is cheaper. And 1 year later is again cheaper. And it's just the price stays stable because they get more profit. So, but the production process will be cheaper and cheaper with the year. So, and let's talk about chromatography, because in this point, a little bit important to understand what does it mean? Because I told you that this is a separation process. And here there is also again some animation. So I try to make, okay, so here. So you have like a stationary phase. So you have like a column. In this column, you have some phase, some solid phase. And for example, here you can see Larry, but not Larry, of course, it doesn't look like this one. But you have like a different molecules. Here you have some molecules, yellow, blue, red, and black. Then you go with a mobile phase. In this case, it can be solvent. Sometimes it can be even water. And you can see that if it goes through this stationary phase, at the end, you have this all of this mixture product. You have a separated. So it looks like a filter. You know, it's a filter with just with some. Sometimes you can even see that you can, for example, take one column, put like a sand in a column, just a sand, and for example, put Coca-Cola, and you will see that at the end you will not get a Coca-Cola the same color as you have on the top, because sand is also a stationary phase, and some molecule will be catch it on the like here you have seen on the video. So and this is like column works. Here you can see also on some pictures that also you can separate. So chromatography, it means separation. It means separation of mixture some compounds. So in our case, I will explain later, this is the similar compounds, but with a different position in a 3-layer in a stereochemistry. And if we are talking about, because for me as a chemist, columns, you use in a laboratory always the columns, because it's the easiest way. You know, it's like you make a soup, and then at the end you just would like to have a meat. So, and you put all of the soup from this column, and you take a meat. So I have like now boiled meat. That's enough. So this is the same what usually chemists makes in a laboratory, because for us it's very important to get this molecule, and at the end you have like some mixture of different molecules to make such a column. Always it's a different how high is the column, what's the stationary phase inside, and so on, what's the mobile phase. But at the end you can always find the condition that you can get separated these compounds, and you have these compounds, and you say, oh, very nice, I have a compounds. Let's test if this compounds against HIV. But if you are talking about production, to make such a column, you can see here, I showed the picture, how it looks in production. And I can remember as I work as a scientist at Merck in Darmstadt, we have really one column, it was 25 meters. It's a 25, it's a huge columns with so many stationary phase, and with the many process, you can separate, and then you need all of this amount of the phase, throw away, take a new one, and so many solvent, and it's a huge amount of a solvent, huge amount of a stationary phase. At the end, it's very expensive. But pharmaceuticals companies at the start do this process because they would like to get medication, as I thought, as fast as possible on the market. So this is why if we go now to, for example, to Cipla and say, Cipla, why you make, I don't know, abacavir? And they say, look at, I don't want to make abacavir with the chromatography because this process is not patent, and this is an old process, because it costs so many money for me that I cannot sell for less money as an originator, and originator has a process which already patented in this country. So it means I cannot sell abacavir, for example. Now you can sell, but at that time you could not sell abacavir because patent process. So, and sometimes it's still be a barrier because of the pricing. So now let's talk about this stereochemistry. So I just want to show you how does it look, lopinavir, in a reality, because always we see 2D, and now you can see on the right, this is 3D. So you can see if you have this bolt, for example, here with NH, this bolt, it means this is, you have a 2D, and it means that this is bond go up. And if you have a like this bond, so it means go down. So we try to put on the 2D two-dimensional picture to understand how does it look on in 3D. And it means that this, for example, lopinavir looks exactly this one, so has exactly this fixed angle, and this is important to have this fixed angle. It's not like a 2 degrees or 20 degrees less, or it's exactly like, look here, because, for example, lopinavir, what is it? It's an inhibitor of protease. we have I show you all of protease inhibitor here, lopinavir, darunavir, and atazanavir. And all of them looks like interesting art objects. Yeah, so it's because we have our protease. Here you have seen, you see protease. And if you see this protease, in the middle, we have like a hole. So this is hole. If, I don't know, do you know what protease make? For example, we have in body many protease. So, and protease, usually if you have a, like a fats or like a long amino acid chain, so it goes through this hole and it cut it in smaller. So it means if you eat some food, this food will be at the end, all of these chains will be cut in smaller, so that your body can take the small molecule easily as a huge molecule. So this helps us to take food inside our body, this protease usually. And the same protease help also HIV virus because HIV virus, I don't know if you will have this course about HIV and science. So HIV virus, there is a integrase, there is a remote transcriptase, and there is a integrase. And for example, protease, very important to have because if viral make with RNA and in DNA make a big chain of proteins, this protein should be cut to put and packed in HIV viral. So this is very important block in that the virus get born the new virus of HIV. So, and of course, we see that this hole, it's like a working hole. So it's a hole which cutted long chain proteins. So it means, what does it mean inhibitions? It means that I block this hole exactly with my molecules. And this is why they like this one, because exactly like this one, I need to put in this hole. It's like, it's like a key I put in a keyhole or in locker. So it's exactly this key, put exactly in this keyhole, in key hole, and it means that I block, and then other proteins can. It means I inhibit protease, and new variant of HIV cannot be born. So this is how they work. And I have here one videos again. I mean, you can also find such videos, many videos in the internet, because pharmaceuticals companies make always these videos, especially if they make new HIV drugs. They always need to explain how it's beautiful to inhibit that one and that one. But from our side, it's very interesting to see because they make an animation. Exactly now, for example, they explain how make HIV make a reproduction of themselves in the cell. Okay, you see here, for example, you have exactly you have a here proteases, you have a here integrase, and you have a transcriptase. And here we speak about again about protease, and you can see that this protease has like this hole. And what's happened in the cell? In the cell, you got this exactly this proteins at the end, and they are long chain proteins. And exactly this protease, exactly what I explained, cut it in smaller one. And why you need the smaller one? Because the smaller one, together with RNA and other proteins and integrase, will be packed in one virion, and this virion leaves the cell, and new HIV virion born. So this is the normal process of reproduction of HIV. And exactly here you can see they show again protease, and they try to show that there is a like a, I don't know which here exactly a drug, but let's say about lopinavir. They show there is in the protease, like here you have seen some active, like here, just a moment, exactly with the light. Always in each protease or in each integrase, in each, I would say in each biological ferment body, you have a special active site. So it means this is a site where usually you have for example, here cutting process. So it means this lighted, how to say, fragments or protease make whole work. So it means if I put my key exactly in this position and block this position, it means this protease cannot work. And exactly what's happened here, exactly, will show exactly that there is a different molecules. For example, they compare one molecule with other molecules. And you know, here they show that it goes inside in the protease. And of course, if it's inside in protease, it makes, if it's exactly like a key, exactly fits in this lock, you have a new bonds and you block. So you block, and you can see later. For example, here you block. So and then protease try to work and try to cut. For example, some proteins cannot cut. So it means you block all this reproduction process. So, and of course, for example, in this video, they explain that, for example, protease, of course, can have a mutation. And it means that if they have a mutation, it means you have seen this, active position of protease. It means this active position, they change a little bit the position, they change a little bit C-base structure and so on. This is why you need to use, for example, instead of lopinavir, you need to use darunavir. Why the lock has been changed a little bit and you need a new key. But at the end, it's a similar key, it just like with small differences. So here was a very old in Ingelheim. It's not in HIV production, but at that time in the 90s, Berlin Ingelheim was in HIV production. But I mean, such a videos you can find many which explain how this molecule, exactly this molecule, works in HIV and what's will be inhibited and what's important and why it's like important to have such a structure. And this is why, if you remember here in our process, this is structure. This is like a mix. This is ratsomat. Ratsomat, it means always mix of different. So you have this structure, but you have a for example, this up, this down, this left, right, and you have so many different 3D structure. But at the end, it's abacavir. But we know that only abacavir with this 3D structure can inhibit. So it means we need to make, for example, on this stage, we need to make separation and get exactly this enantiomer. It calls enantiomer. So it moves like an optical, usually you say it's optical active. So because you have a special angles, and if you go with the light, you will have a optical activity of this duct. So here we are, and I think it was for the production of HIV drugs, part number 1 of APIs. And if you still have a question, you can ask. And for Friday, we will continue. So we will continue with production of finished dosage form. I think it will be usually the second part more interesting for the people because tablets you understand, because here it's too much chemistry. I can understand it's a little bit dry, but on Friday we will speak exactly about pills, about tablets, and this is more understandable and exactly there in this part, because on the part of production of API, we don't have a generics, we don't have an originator, we have a substance. For me, as a chemist, I have never heard before generics. For me, hey, what does it mean with generics? And then as I start to understand 20 years ago in the production process and so on, I understood, okay, generics, it means different production of finished dosage form. But at the end, you use the same API here, here, and here. And this is why important always to distinguish APIs and finished dosage form. And this was the first part with the API, and on Friday we will have a finished dosage form. So we still have a time for the question. Maybe Greg, I see that you have a mic
I
on.
only have a comment. I want to say that I'm a biologist by education. It was super interesting. You brought me back 25 years back to my university.
Look at, oh, nice.
Younger, so thanks. It was fascinating. Thanks a lot. For me, it was not dry at all.
Okay, okay, great. Thank you very much. So do we have questions?
I agree, absolutely.
Okay, good. Okay, I mean, we will have anyway on Friday. Maybe you need a time a little bit to sleep with this information. And I can remember that also if you do it the string and one day after lunch, you get always more questions. So maybe after these 2 days on Friday, we will have more questions and you will bring some in your head and we'll ask some from the one part. It's also okay for the Friday. Okay, so I think we can finish on this stage. And Marina, we see on Friday at 12 o'clock.
Exactly, yes.
Our time, European time. Okay.
Yes, for the English session, exactly.
For English session, okay. Okay, exactly. So
thank you very much, Alex. It was very interesting. I think we'll need to study the molecules now. Thank you, everyone, and I wish you a very nice evening. And as Alex,
I forgot. You can see we have such a.
Yeah, I see that.
You remember? Nice from AIDS. What was it? It was in Amsterdam, I think. AIDS
2018. Yes.
Exactly.
I'm still waiting my T-shirt, Alex. I'm still waiting.
We have it, but it's Corona.
Yeah, you send it by post.
Okay. I do it. Okay, don't worry.
Thanks. Okay. Now
okay,
it was.
Guys and girls, thank you very much. And
see you Friday again.
Bye bye.
Bye bye.
Bye bye.